Mesdames, Messieurs, bonjour. Nous avons le plaisir aujourd'hui d'avoir bien sûr avec nous Elaine Fuchs qui va donner son second cours. Le premier a été une magnifique introduction aux cellules souches, partant des problèmes éthiques pour nous entretenir des avancées les plus récentes en ce qui concerne la biologie de ces cellules. Alors aujourd'hui, elle va nous emmener vers ce qui est véritablement son domaine scientifique depuis des années, avec des contributions tout à fait majeures. Et elle va discuter la biologie et la génétique de la peau et des poils.
Merci beaucoup, Professeur Petit, and welcome everyone. Je ne parle pas de français, je parle seulement un peu. So I will speak in English and appreciate your patience. I'll try to speak clearly enough so that those of you who don't have as much English background will be able to understand what I have to say. I don't do as well in the reverse. So I'd like to welcome those of you who were here last Tuesday and welcome those of you who were not here before. And today, on last Tuesday, I spoke a little bit about the prospects of stem cells in general for regenerative medicine. We talked about different properties of the basic properties of stem cells. And today I thought I would focus my lecture on skin biology with an emphasis on some of the work that we've been doing since moving to Rockefeller University now some 5.5 years ago from the University of Chicago. And so the focus, even though I'll be talking about biology and genetics, will be that of stem cells and their lineages in the skin. And for those of you who were here last Tuesday, you'll recognize that I always like to begin my lecture, usually trying to come up with different examples, but to remind all of you in the audience that in fact, both nature and human beings, we are part of nature, have really had a fascination with our body surfaces. It's one of the few tissues and organs of our body that we actually can see and appreciate on a daily basis, and indeed we in various different cultures of the world have continued to adorn upon what nature has given us. And so there's a natural fascination there about skin, and for me it's been an area that has continued to come up with many fascinations, in this case as a scientist. And overall, there are adult stem cells that reside in the skin, and the definition of adult stem cells in general are that they are cells that are able to make and replenish tissues. And in the skin, these cells exist as slow-cycling progenitor cells, and they are marked by two keratins that my laboratory cloned and characterized many years ago when I was a starting assistant professor, keratins 5 and keratins 14. These cells then give rise to what we call rapidly proliferating progenitor cells. Sometimes they're referred to as transit amplifying cells, cells that have a limited number of cell divisions before they go on to differentiate. I prefer to call these rapidly proliferating progenitors as opposed to transit amplifying cells, because transit amplifying, in a sense, conveys a commitment step. And we really don't know in skin biology, and in fact it's a general problem of much of stem cell biology, where the commitment resides with regards to when is the point of no return, when a cell has left its stem cell niche, becomes committed to differentiate to make a tissue. And in our case, the tissues that we're interested in are those of the skin epithelium, the epidermis, the hair follicle, and the sebaceous gland. I point out that the rapidly proliferating progenitor cells are also marked by keratin 5 and 14. So a major issue that my laboratory has focused on is one of morphogenesis. How do you start with a stem cell, in this case a skin epithelial stem cell, and build a tissue? And then if you can understand that process, how do you start with the stem cell and have the capacity to build different kinds of tissues? And I always sense that if I can answer that question in the course of my lifetime, then I'll know a lot about biology. So what about cancer? Cancer is basically an abnormal state of differentiation, an abnormal tissue. These are cells that, and cancer stem cells are cells that are able to make tumors or tissues. And when we think about cancer stem cells in general, which is a very interesting topic these days, it really boils down to the issue of the slow-cycling progenitor cells that are characteristic of many adult stem cell niches turn out to be a very convenient vehicle for the acquisition of mutations which over time are going to be giving rise to cancer. And so even though they have an advantage in that these are infrequently utilized, which is important for stem cells, they also are long-term survivors. And so this turns out to be, over time, a deleterious, potentially deleterious situation. And if the various different mutations that are going to give rise to cancer do not affect the slow-cycling progenitor status of the stem cell, then there are likely to be a very few number of so-called cancer stem cells that are giving rise to the tissue. On the other hand, if the overall feature of slow-cycling progenitor status is not an essential feature of a stem cell, then the acquisition of mutations may compromise the quiescence of the stem cell without compromising quiescence, and that could give rise to effectively many cancer stem cells residing within a tissue. And so overall, as scientists, we view cancer stem cells and the number of cancer stem cells as basically being predicated on the extent to which these various different mutations compromise quiescence and the degree to which quiescence is an essential feature of a stem cell. And so I'll try to touch upon those questions today at a molecular level. So why do we use skin as a model system for our study? Well, there are several very simple reasons. This is a readily accessible tissue, as I alluded to already, and it turns out that its epithelial lineages are spatially and temporally well defined. In addition, all of these cells that you see in blue, the skin epithelium here, are basically derived from a common embryonic skin progenitor cell, an embryonic skin stem cell. And in the adult, these tissues undergo constant rejuvenation and repair. Your epidermis is constantly exposed to a variety of different physical and chemical traumas of the environment, and as a consequence, it's necessary to have a continual replenishment of cells on your surface of your body. And in fact, every 4 weeks' time for human, you've got a brand new epidermis. It undergoes turnover that rapidly. So finally, there are many different types of epithelial cancers, and they include the most prevalent cancers in the world, the basal and squamous cell carcinomas. So just a few words then about the overall homeostasis and structure of the epidermis and its appendages. The epidermis is a stratified squamous epithelium. It's separated from the underlying dermis, as is its appendage, the hair follicle, from a basement membrane that's contiguous around. And its cells that are attached to the basement membrane turn out to have proliferative capacity. These cells, as they exit the niche and move outward toward the skin surface, undergo a commitment to terminal differentiation. They're eventually sloughed from the skin, continually being replaced by interlayer cells moving outward. The appendage, which is often called the pilosebaceous unit because it includes the sebaceous gland, which is basically an appendage of the hair follicle, is a very different structure, but it shares many of the same characteristics and also undergoes constant turnover throughout the course of your lifetime. At least if you're lucky, it does. It undergoes periods not only of growth of the hairs, but also or of rest of the hairs, but also of growth of the hairs. So another point I'd like to make is that the dermis is very complex. It's composed of fibroblasts, blood vessels, connective tissue, and immune cells, which are also very important for us to fight off infections. And every time you see blood in a wound, you've gone completely through the epidermis. It has the thickness of about cellophane. So every time you see blood, you're basically down in the dermis, and it's very easy to remove the epidermis. So where are the stem cells in adult skin? Well, at the time we began focusing on this question, back when we moved to Rockefeller University, research at that time had accumulated, but not so far as to really understand where the stem cells existed within adult skin in vivo. And so what we knew about epithelial stem cells were twofold, really. We knew that they should be relatively infrequently utilized and therefore slow cycling. And we knew that, like all cells with proliferative capacity that exists within the skin epithelium, these cells should be marked by keratin 5 and keratin 14. So that's not very much information, but using that little information, we came up with a strategy that allowed us to tag and mark the stem cells and ultimately isolate and characterize them. And the tag is the following. We engineered a bitransgenic mouse. One mouse expressed a tetracycline transcription regulatory factor under the control of a keratin 5 promoter, and the other mouse expressed a tetracycline regulatory element driving the control of histone H2B GFP. So now, in the presence of both transgenes, the mouse is green because the transcriptional activator binds to TRE, expresses histone H2B GFP, and if we take a cross-section of the skin and look at it, we see that all of the skin epithelial nuclei are marked green, but only the skin epithelial nuclei. And that's important because in the discussion, as we learned from my discussion with Christine last week, there are about 20 different cell types in the skin, and we wanted to specifically target only the skin epithelial cells. And I point out that there are other stem cells that exist within the skin as well. So the reason why all of the nuclei are green, even though some of the cells only express keratin 5 and 14, the cells attached to the basement membrane, is the following, and that is that histone H2B GFP is very stable, and if we're expressing in the absence of tetracycline constitutively the gene, then we see all of the differentiating cells of the skin epithelium as well as the proliferative cells marked by green nuclei. So now what happens if we simply start adding tetracycline to the animal's diet? Well, now in the presence of tetracycline, the transcriptional activator can no longer bind to DNA, the gene is shut off, and now if we wait long enough, the dividing cells, all the rapidly dividing cells are going to dilute out the histone H2B GFP. Every time the cell divides, there's going to be half as much of this fluorescent protein. All of the differentiating cells are going to turn over in the skin, and so they're going to be lost. And what we're left with is simply those cells that are marked by keratin 5 and keratin 14 and that are long-lived and label-retaining, slow-cycling, infrequently utilized. And those cells reside in a region known as the bulge, and it's present in every single hair follicle of the body, and it's located in a region that is residing just below the sebaceous gland of the hair follicle. These are the sebaceous glands here. And this niche then is the resident niche of these cells. But are these really stem cells? In fact, that's an open question until we do further experiments here. And so now, what kind of experiments are we going to do? Well, the first issue is, why would you bother having a niche of infrequently cycling cells located here within the hair follicle? What's so significant about this as a possible niche for stem cells? Well, if you understand a little bit more about the hair cycle, then I think you'll begin to appreciate why this is of particular interest. So, as I mentioned, in the course of your lifetime, if you're lucky, your hairs not only undergo periods of growth, periods of rest, but also periods of growth. And in morphogenesis, the hair follicle begins by epithelial mesenchymal interactions. There are specialized mesenchymal cells that interact with a single layer embryonic skin multipotent stem cells and stimulate these cells to start growing downward and begin to form and eventually form the hair follicle. The mesenchymal cells become what we call the dermal papillae cells. They're a permanent portion of the hair follicle, and they reside down here at the base of the hair follicle, and they're surrounded by very rapidly proliferating cells that we call matrix cells. These you might think of as the so-called transit amplifying cells, and in fact this we know now is really a compartment of transitly amplifying cells. They will end up being lost at the end of the hair cycle. Once the follicle matures and has made its sebaceous gland at the very end of embryogenesis and basically has fully matured, fully grown downward, now the matrix cells start to also differentiate to create the hair shaft and the channel called the inner root sheath that surrounds the hair shaft. It's also surrounded by an outer root sheath that's contiguous with the epidermis. And so now the hair grows for a while, and then at the end, all of a sudden, there's an exhaustion, possibly of the proliferative capacity of the matrix cells, and now the follicle ends up destroying itself. But it's only the bottom 2/3 of the follicle that ends up undergoing this destruction, where the entire lower part of the follicle, including these transit amplifying matrix cells, are lost. But now, surrounded by a basement membrane, and just attached to it is the dermal papillae cells on the outside. They are dragged upward until they come to rest right at the base of the permanent portion of the hair follicle. And at that base is basically exactly where this bulge resides. And so now I think you can begin to appreciate that if you look at the two stages in the hair cycle, where it turns out that there are mesenchymal interactions with the epithelium, they exist in early hair follicle morphogenesis, which in response, the hair follicle grows downward, and they exist in this resting phase of the hair cycle, at which then there eventually is followed by a new growth of the new hair follicle. And this process is continuing constantly throughout your lifetime. It's really a wonderful system for biology, because how many other systems do you know where the organ undergoes constant degeneration, regeneration, degeneration? So it's a great system to look at stem cell biology. So now you can appreciate why we were interested in seeing this particular residence of label-retaining, slow-cycling cells, and now we wanted to know, are these really hair follicle stem cells? And this then just summarizes what I've just told you. So mesenchymal epithelial interactions are important in somehow stimulating the new hair follicle to grow. That's a clue. And another clue is that this particular niche is coming into contact with mesenchymal cues in doing so. So now we can simply look at what happens to these label-retaining cells during the initiation of the new hair cycle. So here's the new hair germ that's growing down. It's just had its contact now with the dermal papillae cells, and look what's happening. This is marked by Ki67, which is a proliferating nuclear antigen, marking proliferative cells. You can see that these cells are rapidly proliferating, both by this antigen and also by the fact that they no longer have histone H2B GFP label. Well, they do, but it's much lower. And so what we're learning is that as stem cells exit their niche, they start rapidly dividing and dramatically lose the label. What we also know from this study is that only a very few number of so-called label-retaining cells are activated with each new round of hair growth. So you might think that all stem cells become activated and exit the niche. That clearly is not the case. Only a few of them do, because most of these cells retain as much label after the new round of hair growth as beforehand. And that really is suggestive that only a few numbers of hair follicle stem cells are necessary in order to regrow the hair follicle. Not all of them are used. So now what happens in another situation? Let's take a look at a wound response. So in wounding, as I mentioned to you, when you see blood, basically you've damaged completely your epidermis. You're down into the dermis. And so it doesn't take much. And in fact, a scratch will, or a scrape will basically remove the entire sheet of epidermis. So if you do that to a mouse on a very small scale, what happens is the following. So here's the wound site up here. Now you can see that there are many fewer label retaining cells that are left within the niche, and you can see many more of them moving upward. They move along the basement membrane and move upward toward this wound site. And you can see that they do start dividing, but they're also migrating at the same time that they're exiting the niche. So these data suggest that this residence can both leave and refuel or re-epithelialize the skin in a wound response, and it can also move downward to generate the hair follicle. But are, in fact, these cells multipotent? You could argue that they're simply a bag of stem cells, some of which move upward to make the epidermis, and others of which move downward to make the hair follicle. Are these cells really multipotent? And when you think about how would you experimentally demonstrate that, the only way in which you can demonstrate that is by clonal analysis. Can one stem cell have the capacity to do both of these different things? And so we can take advantage of the fact that we can now use fluorescence-activated cell sorting, and that we can also culture these cells in the laboratory. And so we can culture the label-retaining cells, and we can also culture their cells with much less fluorescence, with maybe 100-fold less fluorescence, their progeny, and take a look at what happens. Well, first of all, if we just look at the colonies, what we see is that the colonies from the label-retaining cells tend to be composed of large colonies, composed of small, relatively undifferentiated cells. And on passage, these cells can be passaged for multiple passages, suggestive of a cell that you might predict being a stem cell. In contrast, the cells from the progeny tend to give rise to relatively smaller colonies composed of larger, more differentiated-like cells, and on passage, those cells tend to undergo senescence, what you might expect for transit amplifying or cells with a limited number of cell divisions. So now let's take a look at doing the clonal analysis. And here we can again take advantage of the fact that we can culture these large colonies in the laboratory. And so if we seed them at low density, pick individual colonies derived from single bulge stem cells or bulge putative stem cells, and we now are taking them from our H2B-GFP tagged mice. And so in this case, we're simply culturing the cells in the absence of tetracycline. So we have these green colonies. We now mix them with unlabeled dermal cells and then graft them onto the backs of nude mice. And nude mice are immunocompromised because they're missing a transcription factor that is necessary not only for T cell development, but it's also necessary for hair follicle development. And so conveniently, these animals have no hair. And so the first experiment we want to do is the control of what happens when we just have dermal cells there. And the nude mouse can repair its own wound. So this was the wound, and the nude mouse has repaired it. And in fact, that tells you right away there have to be other sources of stem cells that exist within the skin. That's a good thing, because for those of you whose hair follicles don't undergo constant cycling throughout your lifetime, you wouldn't want to have your ability to repair wounds dependent upon your ability to make hair. And somehow nature has worked that out for you many centuries ago. And so there are stem cells that reside within the epidermis. But now if we do the test experiment, here you can see that when you combine it with now cells that are derived simply from one single label retaining cell, you get these nice tufts of hair. The hairs are green, so they're suitable for bars in late night Paris. And in addition, when you take a cross-section of the skin, you can see that there's green epidermis, sebaceous glands, and hair follicles. And the fact that all of these structures are derived from the derivatives of one single label retaining cell tells you that these cells that reside within the niche have multipotent capabilities. These are multipotent stem cells. So I talked a little bit about this last week, and I will only refer to you the paper that discusses it here, and simply to say that we've gone on to test the potency of these stem cells, and we did so by nuclear transfer experiments. And in nuclear transfer experiments, here one takes the nucleus of a purified bowled stem cell and introduces that into an enucleated mouse oocyte that's been unfertilized to make a diploid cell, which now, if you make a small embryo and implant that into a host mother mouse, what you get are cloned mice that are all derived from that single label-retaining stem cell. Now, in this case, it's a hybrid that you need to make that mouse. It's derived from nuclear transfer experiments. But it's surprising because the fact that we can get mice and that those mice can give rise to normal, healthy offspring, even though they do so at a very low frequency, really tells us that the entire chromatin can be reprogrammed sufficiently to act as if it was an embryonic cell able to differentiate into all 220 cell types of the body, and do so in a fashion that, at least on the surface, appears to be normal. And we see no signs of tumorigenesis or alterations, as there was originally with the first cloned animal, and that was Dolly many years ago. The field has really advanced greatly since then, and it still stands despite the fact that scientists have recently learned how to take skin cells and convert them into embryonic-like stem cells. It's still the one example of using nuclear transfer as a system of generating effectively unprogrammed cells that entails no genetic manipulation of the chromatin in order to do that. In this case, it's exposure to a tremendous mouse oocyte cytoplasm that has this capability. But it's not genetically manipulating the DNA. So in the skin, these cells then are cells that just differentiate into making sebaceous glands, epidermis, and hair follicles. But there are additional stem cells in the skin, as I alluded to. And in fact, what we've known, not only do nude mice repair their wounds, but in fact, we've known from the early studies of Chris Potton that there was postulated to be an epidermal proliferative unit, stem cells that are a subset of cells residing within the dividing layer that's attached to the basement membrane. And somewhere in here, Chris Potton hypothesized that there were stem cells. Fiona Watt's group took human epidermal stem cells in culture, took advantage of the fact that epidermal cells could be propagated endlessly in culture, or fairly endlessly in culture, and she purified cells that existed within cultured populations that had a greater proliferative capacity. But whether or not this correlates to the in vivo situation has been a long-standing open question, and in fact, to date, we still don't have any genetic evidence that would say that one cell within the basal layer of the epidermis is truly different than another cell. And it's really an open question of whether all of these cells are effectively unipotent stem cells, or whether there really is a population of so-called transit amplifying cells that exist within this gamish of a very small number of stem cells. So that's still an open question in skin biology. So the best evidence that tells you that there's really a discrete population of stem cells that exists within the epidermis comes again from lineage tracing experiments. And this is an experiment that was done by Bruce Morgan and his coworkers several years ago. And they made a mouse that expressed the Cre recombinase under the control of the sonic hedgehog promoter. And they mated that to a ROSA26 generic gene that's expressed in all cells of the body, a ROSA26 locus that has a stop codon that exists between a bacterial beta-galactosidase gene and the promoter for the ROSA26. And so that it's flanked by Flox sites, which then can be recombined out when a cell is expressing Cre recombinase. And this is a standard method now that researchers in the field use for lineage tracing analysis, for determining not only where the cells are that are expressing sonic hedgehog in this case, but then where are all of its progeny that are derived as a consequence. And sonic hedgehog turns out to be a gene that is very important in the earliest stages at which the hair follicle is formed. So if you take a look at the lineage tracing from this experiment, what you see is that early developing hair follicle is positive for sonic hedgehog and its early derivatives, whereas the epidermis, here marked only in brown by a keratin gene, is basically negative for blue. And now if you follow it down to look at the hair follicle, the entire hair follicle that's developing, including the bulge, is all blue, whereas the epidermis remains negative. And that really tells you that there are two different populations of cells that are defining these two structures. But now if you wound the skin, analogous to what I told you about previously with H2B GFP, you can see now blue cells within the epidermis. So in a wound response, the hair follicle stem cells contribute to the repair, but otherwise, in the normal homeostasis, that flux of epidermis that is continuing throughout the course of your lifetime is fueled by stem cells that reside within the basal layer of the epidermis. And similarly, we know that there is a population of resident stem cells that exist within the sebaceous gland. And so here, this is work done by Valerie Horsley in my laboratory. We were interested in a particular transcriptional repressor called BLIMP1. And the reason was that it came up in a genetic screen that we did that I won't go into. And BLIMP1 was first identified as a transcriptional repressor of the c-Myc gene in B lymphocyte development. And it turned out that BLIMP1 marked just a population, a very small population of cells right at the base of the sebaceous gland. And in this case, we did a similar lineage tracing experiment, but using BLIMP1 Cre and using YFP rather than LAC-Z. And when you do that, what you find is these BLIMP1 positive cells give rise to the entire cells of the gland. And so these data tell us that these are really resident stem cells able to give rise to the sebocytes, which produce the oil that is a consequence of the sebocytes' destruction. So when these cells have matured, they're destroyed, they release lipids, and those lipids go straight into the hair canal and up on the surface of the skin. That's where the oil comes from, on the surface of your body as well as on your hair. So now if you do a slightly different experiment, and that is to take BLIMP1 driving GFP in a mouse, and you purify those cells and place those cells into culture, these cells give rise to sebocyte colonies, and they contain additional BLIMP1 positive cells, suggestive that there's self-renewal of these cells going on in tissue culture. So for all of these data, we know that there are resident stem cells. Now what happens when those resident stem cells are missing? So now we've done a conditional knockout of BLIMP1. And if we knock out BLIMP1, what we see is that the gland gets bigger. Now that's kind of unusual. Why should the gland get bigger if these are resident stem cells and we're losing resident stem cells? Well, it gets down to this issue that BLIMP1 is a repressor of c-Myc, not only in B lymphocytes, but also in the sebaceous gland. And so in the absence of BLIMP1, c-Myc is upregulated by a great amount in the knockout cells, conditional knockout cells, relative to the wild-type counterparts. And so now what we know from Fiona Watt's studies is that if you elevate the level of c-Myc in the skin, what you see are enlarged sebaceous glands. And in fact, they give rise ultimately to sebaceous gland tumors. And so this was all very satisfying until we began to realize that in Fiona Watt's studies, Fiona showed that c-Myc really controls the transit amplifying population of cells, and those cells proceed to differentiate. And so if we simply activated Myc, maybe the gland should get bigger for a while, but then it should shrink when we've exhausted the stem cells and we've exhausted all the transit amplifying cells. That doesn't happen. And why doesn't that happen? And it doesn't happen for a very interesting reason. It doesn't happen because the bulge, which is sitting below the sebaceous gland, is now superactivated in the absence of BLIMP1. So here are these resident stem cells sitting there up there, fueling the gland, but now they're missing. And what happens now is the bulge, which doesn't express this transcription factor, gets activated, starts proliferating in response, moves upward, and fuels the stem cells of the sebaceous gland. So just like in the wounding, when you wound the epidermis, now effectively if you remove the resident stem cells, you've mobilized the master multipotent stem cell reservoir to do something that it otherwise is not going to do. And so that's an important take-home message. So what are we looking at with regards to the stem cell population in the year 2008? We're looking at this as a residence of infrequently cycling, undifferentiated, keratin 5, keratin 14 positive, multipotent stem cells that in the response to environmental changes, there has to be something to get these cells to do anything. In response to a wound, so if you remove the epidermis, these cells now start to get stimulated. Somehow they exit the niche, move upward, and replenish the epidermis. If you lose the BLIMP1 positive resident stem cells, likewise, these cells get mobilized and they stimulate gland formation. But normally what these cells do is respond to mesenchymal cues at the beginning of each hair cycle, and that stimulates the downgrowth of these cells to make a new hair follicle. And that was effectively their intention, but they've been then utilized in these other very interesting ways that say when responding to injury, cells have figured out a way over evolution to respond to that and try to rescue the situation. And that's really how we're viewing the existence of adult stem cells and why they're used. They're necessary for constant replenishment of those cells that are undergoing apoptosis, cell death, but they also have to be able to repair tissues as best as they can in a wound response. So what do we now know further about these cells and what can we do with these cells? Well, obviously we'd like to know what makes these cells special. Why are they stem cells? What are the unique properties of these cells? And so now we can again go back to our advantage of being able to purify these cells now. And we also take advantage of the ability to very easily separate the epidermis from the underlying dermis and hair follicles by an enzyme. And so we can just discard the epidermis whenever we feel like it. We can take the dermis of our label-retaining cells, make a single-cell suspension, and now run this through a fluorescence-activated cell cycle activator, sorter, and we can separate these cells and take the bright cells and the less bright cells, the progeny, and now instead of using these cells for culture, we basically can go directly to microarray profiling and determine what are the genes that are expressed in the label-retaining cells, the stem cells now that we know are multipotent stem cells, versus their progeny. And so we've done this, and in fact we've used two different methods, and there's another group, George Katsarelis' group at University of Pennsylvania, who's also done similar studies using yet a third method, and all three methods have given us a whole long list, in fact about 150 genes upregulated and 150 genes downregulated in this population of label-retaining cells relative to their progeny. And probably some of these are involved in stem cell activation, some involved in quiescence, some involved in self-renewal, some involved in differentiation. How do we sort all this out? So that's what I'd like to focus on now. And I've highlighted a few of the genes that my laboratory has worked on. I just mentioned that there are all sorts of interesting genes here. Some of them are signaling pathways that are upregulated or downregulated. There are also a lot of genes encoding cytoskeletal proteins and extracellular remodeling proteins. I'll talk about that more next week at our lecture. And And then there are also a lot of interesting transcription factors, and we tend to focus on transcription factors and signaling pathways as a way to learning more about ultimately morphogenesis, getting more to what I find the most interesting questions, which are questions that deal with migration, dealing with cytoskeletal remodeling. So that's what I'm going to focus on today, is what we know about transcription and what we know about signaling pathways. And these are just amongst the papers that we've published just in the last several years. Once you've got stem cells, and once you've demonstrated that these are stem cells, now is when all the fun science takes place, and we can move at quite a rapid pace now in gaining more and more insights. It's like a large puzzle where you start to put together the pieces to the puzzle, and in the first stages it's really boring. You don't know anything, and you can't figure out where all these pieces fit, and all of a sudden you start to see some trees over here, and there starts to see a lake over here, and you begin to really get a sense of how easy then it becomes to putting in, and how much more fun it is to put in all the pieces. So that's the stage we're at now. So what do we know? I'm going to first focus on a pathway that my laboratory has been studying since the early 1990s, and that's a pathway known as the Wnt pathway. I think it's familiar probably to many of you in the audience, if not all of you in the audience. And we were particularly intrigued because there are a number of genes encoding Wnt inhibitors that exist in these stem cells. So for those of you who haven't heard of Wnt signaling or don't know about Wnt signaling, there are really two components. And at the heart of the component, one component is beta-catenin, which is a protein that has been implicated both in cell-cell junctions, so it has a role in cell-cell junctions, but also normally any excess protein that's not involved in cell-cell junctions is rapidly targeted for phosphorylation by this kinase, GSK3, and ubiquitin-mediated turnover. But in the presence of a Wnt signal now, this pathway is blocked, leading to accumulation of beta-catenin. And now, if the cell happens to be making, in addition, DNA binding protein of the left TCF family, then there is a bipartite transcription factor that forms between these two, and that leads to activation of downstream target genes. And we've been working on this since before it was known that left TCF was in this pathway. We were working on left TCF on one side of the laboratory and beta-catenin on the other side of the laboratory. And so Walter Birkmeyer with the yeast-2 hybrid screen really set off our interest in Wnt signaling, and we've been working on it ever since. So what do we know about Wnt signaling in the skin? Well, we know that of the 4 TCF left family members in mouse, all of them are expressed in the skin. 3 of them are interestingly expressed in the skin. And TCF3 is interestingly expressed in the bulge cells. And if we take a look at nuclear beta-catenin now with an antibody, what we see is nuclear beta-catenin not so much in the bulge, but in this early hair germ. And that's right at the stage where the dermal papillae cells are interacting with the bulge. So there's something key about the stem cell activation process that involves nuclear beta-catenin, and TCF3 is there, poised to be ready to take place. So now if we make a Wnt reporter gene mouse, which we did and made available many years ago, published back in 1990s, this is called the top gal mouse, and we look at what happens in the hair follicle cycle. What we see is that at the stage where mesenchymal epithelial interactions are taking place, a few cells within the bulge turn blue. And that's the only time we ever saw signs of top gal activation, and we published that a while back, but we never really understood it until we began to look deeper. Again, back in the late 1990s, we made a mouse expressing slightly higher levels of stabilized beta-catenin, and we looked at what happens in those mice now if we mate them with our top gal mice. And now, at the mesenchymal epithelial exchange, the entire stem cell compartment turns blue. Again, telling us that there's something interesting about a change in the status of Wnt-regulated genes that's taking place at the early stages of stem cell activation to make the new hair follicle. And so now the question is, what's going on there? Well, now in 2005, we can start to modulate the levels of beta-catenin a little more finely tuned, and we can just elevate them slightly so that the normal hair follicles grow, they do their own thing, and now we can see, well, what happens when we have just a little bit of excess Wnt's? They go through their resting phase, so this now is the bulge here at the base of the hair follicle right now in the resting phase. But in fact, if we look at what happens with elevated levels of beta-catenin, these cells cannot hold their resting phase as long as their wild-type counterparts. So instead of acting as if they're quiescent at this stage when they should be quiescent, now the stem cell compartments are all firing, triggering a new round of hair cycle activation. And so by elevating the levels of stabilized beta-catenin slightly, these stem cells are getting mobilized to proliferate and differentiate and make a new hair follicle. So that was another clue that we're really dealing with stem cell activation. And we also knew from our loss of function studies and also studies that were initiated in loss of function by Walter Birkmeyer and his coworkers that that the absence of beta-catenin was also deleterious to the hair follicle, and that in fact beta-catenin appears to be, Wnt signaling appears to be important not only for stem cell activation but also for the replenishment or renewal of stem cells. So now we've profiled the stem cells and saying, what happens when we've just elevated the levels of stabilized beta-catenin a bit? We know that this can act as a transcription factor. We have signs that the transcription factor activity is being upregulated relative to our reporter. And yet, what does that mean with regards to what's going on with the endogenous genes? Well, we can, again, purify the stem cells, but now in the presence and absence of slightly elevated levels of stabilized beta-catenin. And what we learn from that is that here's this whole group of cell cycle genes and transcription factors and adhesion matrix genes that are all getting slightly elevated in their activity, their transcriptional activity. And now if we look at, let's take a look at the wild type and see what happens at just when those quiescent stem cells get activated to make a new hair follicle, the same group of genes. And so here, what we're gaining a clue for is that this is really telling us these are critical genes that must be activated in order for the quiescent stem cell to start to move along this hair follicle lineage, to start to make the new hair follicle. And it's now starting to give us a smaller group of genes that now we can start to hone in on and test further. And so that's the kind of experiment that we're doing. And one of those was TCF3 that was obvious. And so we looked at, well, what's TCF3 doing? Because now, at most stages in the hair cycle, TCF3 is there, but Wnt signaling is not. And so TCF3 must be doing something. And what we learned from our recent studies of Huang Nguyen in my laboratory is that TCF3 is acting as a repressor in the absence of Wnt signaling, and it's sitting there repressing many of the same genes that I've just described. So it's acting to repress those genes in the absence of Wnt signaling, and those genes get activated in the presence. But interestingly, when TCF3 is elevated, it represses not only the hair follicle differentiation, but in fact it represses all the differentiation pathways afforded to these multipotent stem cells. So the epidermis, the sebaceous glands, and the hair follicles, all those differentiation genes are completely damped down when you elevate the levels of TCF3. And that's really telling us that this is a very special group of transcription factors where in the absence of Wnt signaling, these cells are really acting to repress terminal differentiation and repress all the lineages that are afforded to these stem cells. So just to summarize then, we know that Wnt signaling plays a role in early stem cell activation and self-renewal, and it's really, in the absence of Wnt signaling, we're really getting suppression of differentiation. But it turns out that Wnt signaling also plays a late role in differentiation. It's never quite as simple as it seems. And so we've known, in fact, and this is what got us into Wnt signaling, that the hair keratin genes are also bona fide Wnt target genes. And I won't go through this evidence. This is now old studies published quite a while back. But we've known that there's a very strong Wnt signal that switches on now down at the base where the matrix cells differentiate to make the hair shaft. And you can't make a hair shaft without Wnt signaling. So you need Wnt signaling to activate the stem cells and say make the hair follicle, but you also need Wnt signaling later at a high level to say differentiate and make hair. And so we learned that a long time ago now, nearly a decade ago, by virtue of what happens if you really start to elevate the levels of Wnt signaling. You get these nice fuzzy-looking mice. They're very cute, and you're just about ready to sell them to a pet store before you realize they develop tumors over time. And those tumors turn out to be just differentiated hair balls. So we went on to demonstrate that the genetics of human tumors, there's a human counterpart, turns out to be due to stabilizing mutations in beta-catenin. So that tells us that too much of a Wnt signal is not a good thing, but it really gives us now a profile. It tells us that undifferentiated stem cells are here at this end of the spectrum. Stem cell activation requires some Wnts. At higher Wnts, we get hair differentiation, and at very high Wnts, we get hair tumors. So all of what I've told you thus far doesn't really deal with the issue of stem cell quiescence, because at slightly elevated levels of beta-catenin, those cells are still able to enter a resting stage, even though they precociously activate the stem cells. So what's controlling quiescence? There must be other signaling pathways. And fortunately, there were two more of them that were upregulated. One of them turns out to be important, and I'll finish today's lecture on that. And turned out that both TGF-beta signaling and BMP signaling, both of which activate transcription factors, different transcription factors of the SMAD family, are both upregulated in the bulge. And we also showed that bulge cells in culture are both transiently withdrawing from the cell cycle in response to either TGF-beta or BMP. So either one of these two signaling pathways could have been good candidates for controlling quiescence, regulating quiescence of the stem cells. Which of them is, and the way in which we answered this question is again to use mouse genetics, and now we did a conditional ablation of the TGF-beta receptor and the BMP receptor, and it's really the BMP receptor pathway that plays a role in regulating quiescence of these stem cells. So here, we did a very simple experiment. We took our keratin 14 Cre recombinase mice that are controlling Cre recombinase under the control of tamoxifen. And we first did a pulse chase experiment, this time with BRDU as a nucleotide tracer, but basically the same experiment I've already described to you with histone H2B-GFP. And we first do a pulse chase experiment, so we label up the bulge cells. And now we say, let's wait till the resting stage of the hair cycle. And now let's take out the BMP receptor. What happens when we remove the BMP receptor now in those stem cells? And what happens is that the cells can no longer hold their label. There's the BRDU labeled control cells, and all these cells rapidly lose their ability to retain label, and they start to make the hair germ. And so this is really the first definitive evidence that we're activating the stem cells, and we know we're activating them because if we do a short pulse with BRDU, these cells start to incorporate BRDU very rapidly while these cells are still dormant in the control. And so we're really activating proliferation simply by removing the BMP receptor at this particular stage of the hair cycle. So now downregulation of BMP receptor activity is necessary for activation, where upregulation of Wnt's was necessary for activation. These two pathways are acting counter to one another. But it turns out that later on, to make the hair follicle, to make the hair shaft, you need BMP signaling. So later they act in concert together to make the hair shaft. And at this stage, they're acting against each other, one to control quiescence and the other controlling activation. So what happens when we knock out the BMP receptor? We start activating these stem cells, but now if they need BMP to differentiate, now we've activated the stem cells, but we don't have BMP receptor, and we get hair tumors. And those tumors are rapidly proliferating, and they express many of the markers that we and others have in the past couple of years identified as being markers of stem cells. So here's a case, getting back to this issue of cancer and cancer stem cells, where we've lost quiescence, but we've got lots of cells within the tumor that are effectively expressing stem cell markers, suggesting there are lots of activated stem cells in these tumors. Are these cells really stem cells? Are they multipotent stem cells still? Now we can do lineage tracing experiments and simply scratch wound and say, well, can these cells, when they're missing the BMP receptor, repair wounds? And the answer appears to be yes, they can. As here's the epidermis is completely gone here, and now we see blue epidermis. So it looks as if, even though we're activating these stem cells, they can still differentiate to make either epidermis or hair, or at least part of hair that can go partway down the pathway. So now I'll just show you that with this profile, what we have are quiescent stem cells with high BMP levels, low Wnt signaling levels. Activated stem cells require a reduction in BMP receptor activity and an elevation in Wnt. Hair differentiation requires an elevation in BMP signaling and an elevation still in more Wnt signaling. And now, if something happens so that BMP signaling goes down or Wnt signaling goes very high, we get hair tumors. And when you think about it, the very mechanisms that we're learning just by studying and focusing on stem cell activation, when mutated, are what are also giving rise to hair tumors. These profiles are matched at those two stages. And I think that's starting to tell us something very important. We're starting to learn why certain pathways are leading to certain types of cancers simply by studying what's involved in activating a stem cell. It wasn't our intent when we started, but it's really staring us in the face that we are now cancer cell biologists as a consequence of studying stem cells. So we're now trying to understand, well, what is it about the dermal papillae cells? Because we know that those mesenchymal epithelial interactions are necessary. And we've worked out methods, and I won't go into those. We've published those about purifying the dermal papillae cells and molecularly profiling those. And we've learned that dermal papillae cells are making a number of BMP inhibitors. We think some of those BMP inhibitors might start giving us clues as to why mesenchymal epithelial interactions are necessary now to tweak those quiescent stem cells to make activated stem cells. And we'd also like to know, well, what's BMP doing? We're starting to learn what Wnt target genes are. What about BMP target genes? What's BMP doing to control stem cell quiescence? And I'll just literally finish on this story, and this story is just about to come out in Cell at the end of this month. And here we noticed in our transcriptional profiling that there's another transcription factor that is upregulated in the bulge that we didn't know about before, called NFAT-C1. And NFAT family members were first identified in T cells and more recently by Eric Olson's laboratory in muscle as playing key roles in regulating the properties of those cells. And so NFATs are very interesting proteins, and we started to look at NFATs, and we, in this case again, is Valerie Horsley in my laboratory. And an antibody to NFAT marks it exquisitely in just the bulge cells. And we showed that NFAT is indeed upregulated in the bulge cells, shown in red here, that we purified. And we collaborated with Lori Glimscher's lab, who works on T cells and is interested in NFAT-C1 for that purpose. And we looked at the knockout and conditional knockouts of NFAT-C1. And the NFAT-C1 knockout hair follicles cannot hold their label. They don't retain label. Their stem cell compartment does not retain label. And in a short pulse with BRDU, they're more rapidly proliferating than their wild-type counterparts. So we've activated the stem cells by removing NFAT-C1, and these hair follicles appear to undergo constitutive hair cycling. So NFAT-C1, in contrast to BMPs, is not necessary later down the road, or doesn't appear to be necessary later down the road. It's only expressed in the stem cell compartment, whereas BMPs are expressed down at the base of the hair follicle as well. And here we're finding that we're just continually activating the hair cycle. It's going to be an interesting thing to look at these mice. Do they ever get bald over time? We have to wait a few years, because right now they don't show any signs of it. It looks like the stem cells are just continually being activated in the loss of NFAT-C1, and we don't know whether they'll get exhausted early or not. And what is NFAT-C1 doing? Well, we can continue to play this game, and also upregulated are a number of cell cycle genes, as I alluded to, in stem cell activation. And CDK4 turns out to be a target for NFAT-C1. CDK4 is not expressed in the dormant quiescent bulge cells, but it is expressed in the activated stem cells. And CDK4 is elevated in the bulge in the absence of NFAT-C1. And if you elevate the activity of NFAT-C1, which we've genetically done in culture, it represses CDK4 expression, and it represses phosphorylation of RB necessary to drive the G1 to S phase of the cell cycle. And so NFAT-C1 is controlling the cell cycle by controlling the activity of the CDK4 promoter as one of probably several targets that it has. And that's starting to give us a clue, because these cells now withdraw from the cell cycle. We're starting to get a mechanism there and the players in place. It's beginning to get easier and easier to put pieces into this puzzle. And we've shown that NFAT-C1 sites are present in the CDK4 promoter, and they appear to be functional. So now, is there a link between BMPs and NFAT? And there is. And so now if we look at the BMP receptor, the wild type mouse, here's NFAT-C1 in the right place and time. Here's the BMP receptor knockout mouse, no NFAT. So now you say, well, is BMP directly controlling NFAT-C1 gene expression? And it turns out it is. If you take BMP and you add that to cells in culture with a reporter that is now the NFAT-C1 promoter driving luciferase, what you see is now a whopping 100-fold increase when you add BMP to the culture medium. So there's a huge transcriptional upregulation. And you say, well, we've known for a long time that calcium is necessary to activate NFAT activity. So if you just elevate the level of transcription, that's one thing, but you have to have calcium there to activate the activity of NFAT. In the presence of activated calcium, there's not much of a change in NFAT promoter activity, so it doesn't do much for NFAT gene expression. That's what BMP is doing. But now if you take a look at what happens when we now use an NFAT reporter activity, that's a different control. That's taking a promoter that has multiple NFAT binding sites detecting activated NFAT. And now you say, what about calcium versus BMP? BMP doesn't do much to move NFAT into the nucleus and activate it as a transcription factor. Rather, calcium is necessary for that. So now all of a sudden we've got yet a new player in understanding this process where calcium appears to be necessary as well. And so I'll just finish then with this model of what we think is driving the transition between a quiescent stem cell and an activated stem cell, that BMP is necessary for NFAT-C1 transcription. Calcium is necessary for nuclear NFAT-C1 activity. It together then represses CDK4 gene expression and basically keeps these cells in a quiescent state. And we're postulating that mesenchymal epithelial interactions release BMP inhibitors, which at some point tip the cell cycle and the stem cell cycle, and that Wints are also involved in this process, and we don't yet quite understand that mechanism. And together then, BMPs, when they're inhibited, then lead to a downregulation in NFAT transcription. There must be some reduced calcium signaling, and we don't know at all how that's going to fit into it, giving rise to loss of NFAT-C1 and CDK4 activation. And cyclosporin A has long been known to be an immunosuppressive drug. It's used widely in clinics, and it's also been known to have unwanted hair growth effects, and we're now starting to get a clue as to just why that is. And there's never been an NFAT that's been known to be expressed in hair follicles. What cyclosporin was doing in controlling excessive hair growth has been postulated by a number of people, but not looking at stem cells, and now it's really suggesting that here we've got these activities where BMPs are acting as a brake on the cell cycle, and that Wints are acting as a promoter for the cell cycle and stem cell activation, and that these two things must get together, act in concert, one being downregulated, the other being upregulated, in order to tip this balance and take these cells out of their quiescent state and activate them. And so with that, I've already discussed the implications for cancer. Just point out that sometimes when we're scientists in the laboratory, we're really, we're focusing on one thing here. We were focusing on the stem cells and stem cell properties, and all of a sudden we're finding ourselves as cancer biologists who really, it wasn't what we intended to look at in the first place, and maybe it's going to end up being more exciting in the long run. And these are the people in the lab that are really responsible for the work. And I'd just like to point out, actually, my French scientist is Géraldine Gauche, who received her PhD in INSERM in Marseille. And Géraldine has been involved not only in the initial purification of the H2B GFP stem cells and their profiling, she's also involved in the TGF-beta receptor signaling work that we just published in Cancer Cell over the summer. And she now has yet another story that she is working on and finishing off. And if you would like to recruit her back to France, you're going to have a lot of work to do because she's on the job market now, but I really suggest that she's a real keeper. She's really done a beautiful job in the lab. Valerie Horsley is another who is, again, is being interviewed and recruited heavily, and did the work that's going to come out this month, and also did the work on the BLIMP1 sebaceous gland cells. And those are really the main contributors of the work that I talked about today. And this is a group of people that's currently in my lab. They're pretty much from all over the world. So with that in mind, I'd like to thank you all for your patience. I realize I've spent a long time. I've kept you here for an additional quarter of an hour, and I've still got energy to address questions if you've got energy to ask them. So thanks again.